Morning. In this video, I'll give a brief introduction about various lipoproteins and apolipoproteins. Lipoproteins are the transport molecules which helps in the transport of non-polar lipids like cholesterol esters and triacylglycerol. Both these non-polar lipids, they form the central core of the lipoprotein molecule, whereas it will be surrounded by a single layer of phospholipids with proteins. This forms the structure of lipoprotein. In this lipoprotein, the protein part is called as apolipoprotein, and we have four major types of apolipoproteins, which are nothing but ApoA, B, C, and E. Okay, so in this video, I will give you a brief introduction about these various types of lipoproteins and the various types of apolipoproteins, so that the individual metabolism, the understanding of individual metabolism will be little more easier. Okay, so first we'll start with the lipoproteins. Based on the protein to lipid ratio, the protein to lipid content of the lipoprotein, the lipoproteins are broadly classified into four types: chylomicrons, VLDL, LDL, and HDL, and these are the characteristic features of the individual lipoprotein molecules. You no need to memorize the entire things present in this table, but you should know 5 facts about each one of this lipoprotein. What are those 5 facts means? To start with, from chylomicron to HDL, the protein content increases, whereas the lipid content decreases. When you move from chylomicrons to HDL, the protein content increases, whereas the lipid content decreases. Second fact, as the protein content increases, the density of the lipoprotein increases, whereas as the lipid content decreases, the size of the lipoprotein decreases. Okay, from chylomicron to HDL, the density keeps on increasing, whereas the size keeps on decreasing. This is the second fact. Third one, the major component of each lipoprotein. In chylomicron and VLDL, the major constituent is triacylglycerol. You can see here almost 80% of the chylomicrons and 50% of the VLDL are made up of only triacylglycerol. So both chylomicrons and in VLDL, the major component is triacylglycerol. Coming to LDL, here the major component is cholesterol. Almost 50% of the LDL is made up of cholesterol. Whereas coming to HDL, here the major components are proteins and phospholipids. So these are the major components of the individual various lipoproteins. For chylomicrons and VLDL, it is triglyceride rich. LDL, it is cholesterol rich, whereas HDL, it is mainly made up of proteins and phospholipids. Okay, so this is the third fact. Next, the fourth fact, the apoproteins present in various lipoproteins. Initially, I said that there are 4 different types of apolipoproteins: ApoA, B, C, and E. Okay, among the lipoproteins, chylomicron is the one which will be having all these 4 types of apolipoproteins: ApoA, B, C, and E. Everything will be present in chylomicrons. Next, coming to VLDL, they will be having one less. The ApoA will be missing in that. Okay, so they will be having only ApoB, ApoC, and ApoE. Okay, so in this, the ApoB, apolipoprotein B, they can exist in 2 different types. One is ApoB100 and the other one is ApoB48. Okay, so chylomicrons usually have B48, whereas VLDL and other lipoproteins will usually have ApoB100. Okay, next coming to LDL. LDL, we also call it as bad cholesterol, whereas HDL is called as good cholesterol. Okay, it is not only that they have contrasting functions, they also have contrasting apolipoproteins. LDL will have only ApoB, whereas HDL will have all the lipoproteins except ApoB. They have a contrasting nature. Okay, so LDL, they will have only ApoB100, whereas HDL will be having all the other types A, C, and E except B. All the other types will be present in the HDL molecule. Next, the last fact which you should know is the functions of each of these lipoproteins. So these functions I will explain you a little more briefly. Okay, so to start with, the main function of lipoprotein is to transport the non-lipids like fatty acids and cholesterol in their esterified form. So that means as triglycerides and cholesterol esters. Both these triglyceride and cholesterol esters can be formed in our body from 2 different sources. One is exogenous diet. Okay, that means from diet. Okay, so which reaches our body or enters the body through intestine. The other one is endogenous synthesis, which takes place in the liver. From these two sites, the lipids will be transported to various parts of our body with the help of our lipoproteins only. Okay, so how it is done, we will see in detail now. So to start with, the dietary lipids, which are dietary lipids means the major dietary lipid, the lipid present in our diet is triacylglycerol. So this triacylglycerol along with few cholesterol, suppose if you are having some non-veg food or some milk, butter, ghee, all those things will have some amount of cholesterol. Okay, so triglyceride is a major component. Along with that, you will be having some cholesterol, fat soluble vitamins, and phospholipids and other things will also be there. So all these from diet will be observed, okay, and it will be packed into chylomicrons and it will be released into the circulation. Okay, so from this chylomicrons, from the triglycerides present in the chylomicrons will then be transferred to peripheral tissues like skeletal muscle and heart. Okay, heart and other tissues, it will be donating, the chylomicrons will be donating the dietary triglycerides. Okay, so from whatever packed in the intestine will be delivered to skeletal muscle, heart, and other few other tissues. Okay, so after serving all these tissues, if there is some excess triglycerides, they will be dumped into the adipose tissue. Okay, so the triglycerides from chylomicrons will be donated to all these tissues. After donating the dietary triglycerides, what will happen means the size of the chylomicrons will shrink, and we call that shrinked chylomicron as chylomicron remnant. Okay, so now within this remnant, what will happen? All the dietary triglycerides were already donated to the other tissues. Okay, so they will be mainly having dietary cholesterol, fat soluble vitamins. They will be having only these two things. Okay, so now this chylomicron remnant will be taken up by the liver and it will be catabolized. Okay, so this is how the chylomicrons helps in transport of dietary lipids from intestine to the peripheral tissues. Okay, so chylomicrons mainly involved in the transport of triglycerides. Okay, dietary triglycerides from intestine to peripheral tissues. Next, coming to the endogenously synthesized lipids. So both triglyceride and cholesterol both can be synthesized in the liver. So all these endogenously synthesized lipids, they will be packed and they will be released into the circulation as VLDL molecule. Now this VLDL molecule, same like chylomicrons, they will be donating the triglycerides to the peripheral tissues like skeletal muscle, heart, and if it is in excess, it will be donating it to adipose tissue. So after losing its triglycerides, so initially what will happen means in VLDL, the ratio between triglycerol and cholesterol will be 5:1 ratio. That means triglycerides will be more, whereas cholesterol will be little less. Okay, so as the VLDL starts giving triglycerides to the peripheral tissues, the triglyceride content will decrease. Okay, and the ratio will become 1:1 to form a new lipoprotein called as intermediate density lipoprotein. So VLDL, it brings the endogenously synthesized lipids from liver, transfer the triglycerides alone to the peripheral tissues, and they get transformed into intermediate density lipoprotein. Now this intermediate density lipoprotein has two different fates. One is like chylomicron remnant, it can be taken by the liver and it can be completely catabolized there. Or alternatively, what it can do is it can further donate its triglycerides to the peripheral tissues. Okay, so further it can lose its triglyceride and it will become a cholesterol rich lipoprotein called as LDL. Okay, so from ideal, if further triglyceride molecules are removed, what will happen? The only inside we will be having only cholesterol. Okay, so such cholesterol rich lipoprotein, we call it as LDL molecule. Okay, so now this cholesterol rich LDL will be taken up by the peripheral tissues and the cholesterol within it will be utilized by it. Okay, so the function of LDL is to transport the endogenously synthesized cholesterol from liver to peripheral tissues. If more and more LDL cholesterol is accumulating in the peripheral tissues, it can lead to atherosclerosis. That's why LDL is called as bad cholesterol. Okay, in order to prevent this, we have another lipoprotein which will be constantly removing the cholesterol from peripheral tissues and taking it to the liver for excretion. That is nothing but HDL molecule, high density lipoprotein. Okay, so since it removes cholesterol from peripheral tissues to take it to the liver for export, it helps in decreasing the cholesterol level. It helps in preventing the risk of atherosclerosis. It is called as good cholesterol. So that's it about the functions of various lipoproteins. Okay, so grossly what we can say is chylomicrons and VLDL, they help in the transport of triglycerides, okay, from the site of synthesis or site of origin to the target tissues. Okay, so what is the site of origin for dietary triglycerides? It is intestine. So from intestine, the triglycerides are transported to the peripheral tissues by chylomicrons. Okay, next, the endogenously synthesized triglycerides from liver, it is taken to the peripheral tissues by VLDL molecule. Okay, next coming to LDL and HDL, they are involved mainly in the transport of cholesterol. LDL transports cholesterol from liver to peripheral tissues, whereas HDL just the reverse, okay, from peripheral tissues to liver. Okay, so that's it about the functions of lipoproteins. So these are the facts you should know about lipoproteins before going into its individual metabolism. Next, we will move on to the apolipoproteins, which are nothing but the protein part of a lipoprotein. As I already said, we have four different types: ApoA, B, C, and E. About each one of these types, you should know again four different facts. What are those four facts means? First thing is where it is synthesized. Second thing, what are the different subtypes of apolipoproteins? Third one, in which lipoprotein it is present. And finally, what is its function there? So these are the four facts you should know about each one of this apolipoprotein. Where it is synthesized, what are the subtypes, in which lipoprotein it is present, and what is its function. So first we will start with ApoA. So this ApoA is mainly produced in intestine. That is the reason it is also present in the lipoproteins which are synthesized exclusively in the intestine. Okay, so in intestine we synthesize actually HDL and chylomicrons. Okay, these are the two lipoproteins which are synthesized in intestine. Okay, so both these will contain ApoA in it. Okay, so this ApoA can be of three different subtypes: ApoA1, A2, and A4. In that, ApoA is the one which has a very important function in HDL metabolism. The other two doesn't have much physiological significance. It is mainly the ApoA1 that has a lot of biological functions, mainly in HDL metabolism. So in HDL metabolism, our ApoA1 has two important functions. One is it activates LCAT enzyme, lecithin cholesterol acyltransferase enzyme, which is needed for storing more and more cholesterol within the HDL molecule. Okay, so thus it helps in the reverse cholesterol transport. Second, it also acts as a ligand for the hepatic receptors for HDL uptake. The liver tissues, they will identify the HDL molecule and they will take up the HDL molecule only with the help of this ApoA1. Okay, so overall it plays an important role in HDL metabolism, okay, which is needed for reverse cholesterol transport. That is why it is called as anti-atherogenic in nature. ApoA1, which helps in HDL metabolism and which helps to prevent the risk of atherosclerosis, is called as anti-atherogenic in nature. Okay, so that's it about ApoA. Next, coming to ApoB. So this ApoB, as I already mentioned, we have two different types: ApoB100 and ApoB48. In this, B100 is synthesized exclusively in liver, whereas B48 is synthesized in intestine. Both in liver and intestine, the ApoB protein is synthesized from the same gene. Okay, but what is the difference means? In the intestine, by a process called as mRNA editing, the cytosine nucleotide somewhere in the middle of the RNA will be edited and it will be converted to uracil. So what will happen means the codon CAA, which codes for glutamine, will be converted to UAA, which is a stop codon. Okay, so stop codon means the synthesis of protein will stop at this point, so that the ApoB48 will be only having 48% of the amino acids than compared to the ApoB100. Okay, that is why it is called as ApoB48. Okay, so in liver, the entire mRNA will be translated so that we get a protein with full set of amino acids. That's why we call it as ApoB100. Whereas in intestine, because of this mRNA editing process, only we get a half of the protein, 45% of the protein only we will be getting. That is why it is called as ApoB48. Okay, so next coming to the lipoproteins, which contains these things means, as ApoB100 is produced only in liver, it will be seen in the lipoproteins which are also produced in liver, like VLDL and LDL, which are produced from liver, will be having this ApoB100. Whereas ApoB48, which is produced in intestine, will be present in chylomicrons, which is also produced from intestine. Next, coming to the functional part, in VLDL and chylomicrons, ApoB protein doesn't have any specific metabolic function. They just act as a structural component. But for LDL, okay, so it acts as a ligand for peripheral cell uptake. Okay, in the ApoA1 we saw that it acts as a ligand for hepatic uptake of HDL molecules. Okay, so liver cells will take up the HDL only with the help of ApoA1. Similarly, the peripheral tissues will take up the LDL only with the help of ApoB100. Okay, so that is the function of ApoB100. Since it plays a major role in LDL metabolism, we say it is atherogenic in nature. Okay, so ApoA1, which plays a major role in HDL metabolism, we call it as anti-atherogenic, whereas ApoB, which plays an important role in LDL metabolism, we call it as atherogenic. Okay, so that's the reason their ratio is used to predict the risk of atherosclerosis. Next, coming to the third type, ApoC. So this ApoC, it is produced mainly in the liver. Okay, it is produced mainly in the liver cells. Okay, so in the liver, it is usually produced along with HDL molecule. It is produced usually along with HDL molecule. It will be produced. So from this HDL molecule, it will be transported to chylomicrons and VLDL. Okay, that's why we say that it is present in chylomicrons, VLDL and HDL. Actually, ApoC will be synthesized along with HDL only. In this, after synthesis from the circulation from HDL, the ApoC will be transferred to chylomicrons and VLDL. So it will be present in all these three lipoproteins: chylomicrons, VLDL and HDL. That means except LDL, it will be present in all the other lipoproteins. Coming to the types, we have three different subtypes: C1, C2 and C3. But among this, the one which has a major function is ApoC2. So this ApoC2, what is its action means? It activates the enzyme lipoprotein lipase, which is needed for the triglyceride metabolism, which is needed for the VLDL as well as chylomicron metabolism. Okay, so ApoA1, A is needed for HDL metabolism, ApoB is needed for LDL metabolism, whereas ApoC is needed for chylomicron and VLDL metabolism. Okay, so the ApoC2, it activates the lipoprotein lipase enzyme which is present in the peripheral tissues. This lipase in turn will hydrolyze, as the name says, they will hydrolyze the triglycerides present in VLDL and chylomicrons, and they will take up the fatty acids. Okay, so thus ApoC2 plays an important role in the metabolism of chylomicrons and VLDL by activating the enzyme lipoprotein lipase. Next, coming to the last type, ApoE. So this arginine-rich lipoprotein, it actually it resembles ApoC in two ways. One is like ApoC, this is also produced in liver. Okay. Second, like ApoE, this will also be produced along with HDL, and then it will be transported to chylomicrons and VLDL. Okay. So same like ApoC, this is also present in all the lipoproteins except LDL molecule. Okay. Now coming to the functions. In HDL, ApoE doesn't have any role, but in chylomicrons and VLDL, what is the function of ApoC? I said it will be helping in removing the triglycerides from chylomicrons and VLDL. After removing the triglycerides from chylomicron and VLDL, it will be getting transformed into their remnants. Chylomicron will be forming chylomicron remnants, whereas VLDL will be forming IDL. So both these remnants will be identified and it will be taken up by the liver for catabolism only with the help of this ApoE. It acts as a ligand for hepatic uptake of chylomicron remnants and VLDL remnant. That means IDL, intermediate density lipoprotein. Okay, so that's it about the functions of various apolipoproteins. Okay, so before going to the individual lipoprotein metabolism, it is very important to understand these various facts about the lipoproteins and apolipoproteins. Okay, so once you are thorough with that, then it will be easy for you to understand the individual metabolism, which I will be discussing in the subsequent videos. Thank you.